All right, so today we're going to continue on talking about the respiratory system. And we're going to begin just by reminding ourselves of some of the different zones and structures that we're going to find in our respiratory system. All right, take another couple seconds on that. And let's take a look. Okay, cool. All right, so when we began our study of the respiratory system last time, what we were talking about was first just some of our pathways. So our airways into the lungs, right? We've got our mouth, our nose, leading to the oro and nasopharynx. Your pharynx continues, moves into your larynx. So we have a split actually between our digestive system going down the esophagus and your larynx, your voice box, leading you into the lungs. Your respiratory system is anterior, so in front of that digestive system. Okay. So as we move into our trachea through some of our smaller bronchi, we see that we have cartilage plates and rings. Specifically, when we're up in the trachea, we'll notice that these cartilage rings are incomplete at the back. They're just C-shaped so that there's room for the esophagus to pass behind so that you can swallow big gulps of food without impacting any of the surrounding structures too much, so that you can have enough space without food getting stuck, basically. That's part of our conducting zone, but not all of our conducting zone. So as we get smaller, the conducting zone is going to be filled with dead air, right? So it's where gas exchange is not happening. So the smaller structures, our bronchioles and our terminal bronchioles, don't have cartilage, but we're not yet to the location where we start to have gas exchange. So it's not until our respiratory bronchioles and then our alveolar sacs that we start to have gas exchange. At this point, we don't have any cartilage left either. But cartilage is in the conducting zone, but not the whole conducting zone. Okay. Cool. So we left off here last time. We had mentioned that the conducting zone is partially for warming up that air, humidifying it, so that when it hits your lungs, it's not too painful. We got a nice environment there in those delicate tissues doing gas exchange. And we have some sort of protective properties in the conducting zone as well. So when we're looking at that epithelium, right, so that lining of our respiratory tract, we have ciliated cells with finger-like cilia We also have goblet cells secreting mucus. And overall, the role is to hopefully prevent little particles, potentially pathogenic particles, from getting into your lungs. Because now I trap them in that mucus and then have you cough it out, basically, right? So actually, when you're coughing out globs of mucus, that's not necessarily the worst thing. That's partially your body trying to protect itself. Okay. So we refer to this structure as a whole as the mucus escalator. Super lovely term, but hopefully pretty memorable. This is what it would look like if you were looking at a histological slide. So here we see those ciliated cells here, and then we'd have to get kind of in between them into glands and some pits in order to get that mucus producing cell layer. Okay. So the point of the respiratory zone, after we get deeper and deeper and deeper past the conducting zone in the respiratory zone, we want to start exchanging gases. So we're trying to take in oxygen, get rid of carbon dioxide. So these gases are swimming around in your blood. In the respiratory zone, we're putting them in really close contact with that air that you've breathed into your lungs. Bless you. Now, oxygen and carbon dioxide are both small nonpolar gases, so we're not going to need any transport proteins or channels or carriers or anything for our gases to be exchanged. They're just going to move by simple diffusion, right? So we are going to think about gradients. We're going to be thinking later on about where do we have high carbon dioxide? How high? Where do we have high oxygen? How high? And the reason we're going to care about that is because we need these gases to move by diffusion. Okay, so diffusion can go directly across those cell membranes, but we go from high concentration to low concentration. So that's what's happening beginning in those respiratory bronchioles, but really where most of this is going to happen is down in your alveoli, which are these little grapes here in these grape-like structures called alveolar sacs. The tubes going into the alveoli are called alveolar ducts. So let's zoom in on one of those. So here we have that respiratory bronchiole. We can see even that we're starting to have some of those alveoli clustered on here. So when you've seen the word alveoli before, you saw it in anatomy lab for like tooth sockets, right? So think about these pits, these little clusters as being kind of sockets, kind of little depressions or holes, right? So we have this duct going in. We've got a big alveolar sac composed of multiple alveoli. So this is where most of our gas exchange is happening. And that can happen because bless you, it's a sneezy day today. We're really close to our blood here. So we have a fine layer of capillaries surrounding like all of our alveoli. This is like completely covered. We've got a big blood sheet covering the alveoli, essentially. Yeah. Now, you also have tons and tons of alveoli. You have 300 million of them. If you were to rip them out and spread them all apart, obviously don't do that. You could cover like the size of a tennis court here. Okay, so tons and tons of membrane for this gas exchange to be happening across, right? We like big surface areas when we're looking for gas exchange, right? It speeds things up, makes it easier for stuff to move across. We're looking at an individual alveolus. An alveolus is just going to be a single one, right? We would notice that it has a little pore, an alveolar pore. So the alveoli are connected to each other, which is good. You want some air to be able to pass from one to the other. Your lung is made up of all these sacs of alveoli. Now, we're going to look in a second at our alveolar cells. So we're going to see 3 types of cells that we're going to be interested in here. We're going to have cells that make up the wall of the alveoles. We're going to have cells that are secreting something called surfactant, which helps the alveoli not collapse in on themselves, basically. And then we're going to have some macrophages. So that was one of our examples of our leukocytes, our white blood cells, right? It was one of our agranulocytes. So here's a concrete example of where you might find. But before we look at that picture, I'm going to look at that relationship in real life between the alveoli and those capillaries. So here you can see those tiny alveoli just totally surrounded by the capillaries. And here they've put basically different colored fluids into the lungs in order to trace out. In red here, we've got the blood. And then in yellow here, we've got our airways. So you can see how they really closely just follow each other, matching up so that we can have as much contact between the blood and the air as possible. So here's a zoomed in cartoon picture of the alveolus with those 3 types of cells. Okay. So most of our alveolar cells are going to be this type 1 cell. Okay. So our type 1 cells are here in yellow. Okay. They're epithelial cells. They're making up the walls of the alveoli. And they're going to be what we're thinking about when we're thinking about being in contact with the blood, right? So here we can see a type 1 alveolar cell. All right. And I picked this one out because we can see it's right next to the capillary with its endothelial lining here, right? So we can see them right next to each other. The type 2 cells are sparser, right? So we have fewer of them. So our type 2 cells are secreting surfactant. Okay, so surfactant, we're going to have this basically kind of watery, soapy kind of stuff that's going to make sure that if your alveolus happens to get small and those walls kind of touch, so they're super flexible, right? You're not going to get them stuck to each other and not able to open your lung again. So that's what our surfactant is going to do. And we'll, in a little bit, look at water tension and surface tension and why that surfactant is important. And here we have an example of an alveolar macrophage. Okay. So one of the ways that we're protecting our bodies from any nasty business we might be breathing in, so that mucus escalator in the conducting zone. Another thing we're doing is if you are breathing in some respiratory illness, right? We've also got some alveolar macrophages here. And you can see the head of one also here. And hopefully noticing that if it happens to make its way into the alveoli. Zooming in deeper here, we are looking at the respiratory membrane now. So these are those same cells that I circled on the image before. So here we have that type 1 alveolar cell, the alveolar epithelium, right? And here we can see it pressed up right against the endothelial lining of the capillary. Right, so epithelium, remember, like lines anywhere you have something that we might consider like the outside of the body, right? Anywhere where we're contacting the external environment, right? And that air that you just freshly breathed in, it was just outside, right? Hasn't crossed any cell layers yet. So that's why we have epithelium in the lungs versus this endothelium, right? Your blood is inside your body. That's why that has a different name, that endothelial cell layer. Okay. So in order for our gases to get across, right? I'm just going to picture, like we can imagine an oxygen molecule here. It would have to cross first the 2 membranes of that endothelial cell, that type 1 alveolar cell. Then we have a layer that's just kind of gluing these cells together. So that's our basement membrane. Right? Our basement membrane here is our glue. And then we're going to have to cross that endothelial cell. Right, so 1, 2, 3. And now we're in the blood already. So now that we're in the blood, we could get into an erythrocyte and get sucked onto a hemoglobin to get carried around. We could run around dissolved in the plasma. Well, we'll get to what happens with the oxygen when we talk about transport of our gases. Okay. So we got a thin layer that we have to go through. Easy barrier for diffusion, right? So just that type 1 cell, the basement membrane, and then those capillary endothelial cells. Really thin, right? So really thin, not hard to cross. That's it. That's our respiratory membrane. Lots of surface area because we have lots of alveoli, lots of capillaries here. Here we can see a different picture of the alveoli. So here we have a bronchiole, not specifying exactly what type here. Right, so that bigger air passageway, but this is probably part of our conducting zone. Then we can see some of those alveoli, those sacs surrounding it. So as we start to talk about like the mechanics of breathing, we're going to want to think about other structures in the thorax, right? And be helpful as we think about breathing to remember back to when we were talking about veins and that respiratory pump. We're going to get in big detail on what's going on with the thorax now. Okay, so just so that we're all on the same page, our chests, when we're thinking about that chest wall, it's airtight, right? You don't want anything coming in through here, right? No big gaps out to the external environment. We've got a bony structure. So we've got our rib cage, we've got our sternum, we have those thoracic vertebrae in the back, right? So making that cage. And then we have muscles that are going to allow these structures to move. So pressures are going to be really important as we think about breathing, just like pressures were important with blood. Okay, so we're going to have surrounding. Okay, I'm going to be lazy with this rib cage, but that's going to be enough ribs for us right now. Okay, so we're going to have external and internal intercostals. Right, so they're going to look like that. And they're kind of cross-hatched, go in different directions on top of each other. Okay. And then we're going to have a diaphragm sitting below. Okay. So the diaphragm is like the floor of your rib cage. So that's going to be the divider between your thorax up here and your abdomen down here. So we're going to see that the way we'll change pressures surrounding the lungs is going to be by using those muscles and some recoil of these structures in order to change the size of the thorax. So as you breathe in, you can feel it immediately, right? Like put your hands anywhere on your rib cage, right? Your rib cage is going to expand as you start to breathe in, and then it's going to collapse as you breathe out. So those are our big structures. Now, when we look inside, before we get to our lungs, I'll draw them on there. Lung. These are very bad lungs. Don't worry about it. Okay. But our lungs have basically punched their way into like a thin little bag. So along the inside of the rib cage, we have a lining called the pleura, and it lines the inside parts of the rib cage. And then because the lungs have punched into it, it has a turn and also lines the outside of our lungs. Okay. So we would be able to trace kind of all the way around like that. So that's our pleura, or the pleural sac. Okay. And in there, we're going to have some fluid. So this intrapleural space, intra is like between, pleural is the pleura that we just drew there. Okay. So that intrapleural space is going to be filled with a little bit of intrapleural fluid. That space is, you're going to see, not going to be very large, right? It's going to have these two membranes mostly pushed against each other, but we got about 15 milliliters of fluid in there. So here's a diagram of that. So here we can see those ribs, the sternum, some of our intercostal muscles, diaphragm here. And then we see that pleural sac surrounding each lung. So as we go through thinking about the mechanics of breathing, we're going to be thinking about what's going on in the lung tissue itself, right? So when we think about this lung tissue, we're really going to be thinking about the alveoli. Okay. And then we're going to be thinking about this tiny little intrapleural space a little bit between the parietal pleura, so that part lining the walls of the rib cage, visceral pleura, this Viscera are just like organs. It's a general word that means organs. So visceral pleura are on the organ, in this case, the lung. Then we'll think about how the muscles and surrounding bones change the size of these locations and therefore change our pressures. So we're going to think about those forces now. So we're going to begin with a question, actually. So we're going to be looping back to some of the same concepts that we were thinking about when we were talking about our cardiovascular system. And so a bunch of those words are going to come back, those physical properties. Okay. So with that in mind, when we think about ventilation, which is physical movement of air, right? When we think about respiration, we're thinking about the gas exchange specifically. Ventilation, the venting is just moving the air. I really think is going to be responsible for that movement. All right, take another couple seconds. Right. Okay. So see why you picked thorax and abdomen, because that was what was responsible for our blood flow, for the veins, right? But now that we're going to be thinking about the air, right? Air is going to be moving from the air in this room into your lungs, and then from your lungs out to this environment that we have in the room. Okay? So all of our airflow is going to be driven by our familiar equation. Flow is equal to a difference in pressure divided by our resistance. Okay, so we're going to need a pressure difference to move the air. And then when we start to think about things like asthma, right? When we start to think about things that are going to make it harder or easier to breathe, we're going to be thinking about that resistance, right? So we're going to be thinking about changing the behavior of those walls of our airways, our pathways. Okay. So we're thinking about thorax versus the external environment. So when we're talking about this air movement, talking about bulk flow, right? So just that F in that equation, we're thinking about the air moving in and out of the lungs for our ventilation, okay? So pressure gradient is driving that flow, right? So when we make high pressures in the lungs, right? You have a high pressure in the lung relative to lower pressure in the environment, you're pushing air out, high to low. You're breathing air in, right? What's happening there is you've created like a vacuum, not exactly a vacuum, right? But you've created a low pressure environment in the thorax relative to higher environmental pressure. So you're sucking air in towards that low pressure. Okay. So we're moving from high to low pressure. In either case, just differs where we have the high, where we have the low. And our formal terms for these two directions, went back on my screen, but not up here, so that's fine. Okay, so our formal definitions for inspiration, that's breathing in, right? So inspiration is when we have low pressure in the lungs, right? So we have a lower pressure in the lungs relative to a higher pressure in the environment. We're going to see the pressure in the environment, right? We don't really change the pressure of the air in this room. It just kind of is what it is, right? Maybe weather could change it. Maybe if you go deep down underground or way high on a mountain, it might change a little bit, but nothing we do right here is going to change that atmospheric pressure. So we're going to see that that's actually going to stay a stable number. So what we're able to vary is the pressure in the lungs lower or higher. So when we make it lower, we breathe in, that's inspiration. When we make it higher, okay, relative to that same atmospheric pressure, we're going to expire, right? So we're going to breathe out, okay? Expiration. All we have to look at, we're going to see lots of lung pressures, but all we have to look at to figure out inspiration versus expiration, breathing in versus breathing out, going to be those two pressures. All we need to know is the pressure in the lungs. We're going to see that by the lungs, we really mean the alveoli here. And we're going to need to know the pressure in the environment, the atmospheric pressure of the place that we're in. And that's it. Just those two numbers, no matter what other variables you see, just those two numbers are going to drive this movement of air. Okay. Now, when you breathe in and out, we're going to be thinking about different lung volumes, different sizes of breath. Your normal resting breaths are going to be what we'll call a tidal breath. But we're not breathing out every single little bit of air that we have in our lungs. We're not completely filling them when we breathe in either. So breathe out as much as you want, but there's still going to be some air left in your lungs, which is good because otherwise they would collapse, right? So that amount that we have left between breaths is our functional residual capacity. So capacity, like being capable, residual, think like extra, functional, it's functioning. I don't know why they put that part in there. Okay. Cool. All right. So pulmonary pressures. These are the various variables you might expect to see in any question about our lungs, right? We're going to be thinking first, and most important, it's going to be that pressure in the atmosphere versus that pressure in the alveoli, right? So when we're thinking about high to low, right? We're looking for which of those two numbers is high, right? That's what's driving that airflow. That's what our delta P is for our flow, right? So that difference between the atmospheric pressure and the alveoli is here. We do have other pressures that from a physiological perspective are important, right? So in terms of making this work, but they're not what's driving the movement of air. So when we think about those other pressures, we're going to be thinking about pressure inside that pleural sac. So we'll have an intrapleural pressure. So that's what the IP is. We're also going to have what we refer to as a transpulmonary pressure. So transpulmonary, we're thinking like all the way across. Okay. So we're going to have a number that compares that pressure inside the alveoli and adds in, really subtracts out that pressure in that intrapleural space to give us a full pressure across the lungs, right? So trans is that across bit, pulmonary, that lungs. We can think of this as the distending pressure across the lung wall. So distending has to do with deformation, so shape changes, right? So what we're saying is that that transpulmonary pressure is responsible for the force that's going to be moving the lungs, right? Either making them bigger or smaller. That's why we care that we have a transpulmonary pressure. So going through each of those, for our atmospheric pressure, let's think about sea level as our example to begin with, okay? So the atmospheric pressure at sea level is about 760 millimeters of mercury. If you go way, way, way high up a mountain or up in a plane, right, it's going to be decreasing as your altitude increases. So as you go higher up, that number goes down. If you go underwater, right, you felt that pressure if you dive, right, increases as you go below sea level. Now, we can give pressures either in absolute values or in relative values. Okay, so what we have here is an absolute pressure, right? So this is absolute. So that's the pressure, like if you measured it, right? So when we're thinking about the lungs, right, we're going to be thinking about like, it's not going to be a real number, right? But the real pressure inside the lungs could be like 800 in absolute values, right? Higher than the pressure of the atmosphere. So you breathe out, right? Number is really way too large. But we're not actually going to give our pressures like that because we can move in different environments. And also then we have to remember the exact atmospheric pressure in a situation. So what we're going to do instead, usually, is give our pressures relative to atmospheric pressure. So we're going to be subtracting out that baseline external pressure, 760. Okay, so this would really be a pressure of, make sure I can do subtraction in my head, right? So this would be like 40 is greater than 0. That's how we would say that in relative terms. This is going to be super convenient because this means that to figure out whether air is moving in or out, whether you're inspiring or expiring, right? If you have a positive number, you're more than 0, right? So if we have a positive alveolar pressure, we're going to be breathing out. If you have a negative number, you're going to be less than 0. So negative numbers for our alveolar pressure are going to mean you're breathing in. Yeah. So this is quite convenient for comparing things rather than having to remember exactly what the atmospheric pressure is. Okay. So that intraalveolar pressure, one that we're comparing to our atmospheric pressure, symbolized by P sub ALV for the alveoli. It's the pressure of the air in each of those little sacs in those alveoli, okay? So we're going to give those pressures relative to atmospheric pressure when we're really talking about lung pressures. This is going to change, right? Your alveolar pressure is going to change, which is why you can breathe, right? So sometimes it's going to be more than your atmospheric pressure so that you can breathe out, get rid of your carbon dioxide. Sometimes it's going to be lower than the atmospheric pressure. You have that negative pressure, you're going to have air move in so that you can get nice, fresh oxygen. So we're going to have phases of respiration where we alternate from having a positive number, negative number, back and forth. Okay? So during inspiration, it's negative. During expiration, it's positive. And that's what's driving the movement of air. So driving ventilation. Okay. We're going to do a couple of questions, just emphasizing this idea of absolute and relative pressures and inspiration, expiration, breathing in and out. All right. Are you ready? Okay. So we're going to start with some absolute pressures to begin with. So let's start with an absolute pressure, the alveoli, so PALV of 764. And so you don't have to memorize it. I gave you an atmospheric pressure of 760. You're going to enter, so inspiration or exit, expiration. All right. Yes. So 764, higher than 760, okay? So we breathe out because we go from high pressure to low pressure. Okay? These are going to be kind of quick, but they're all the same kind of question, right? So what if we have a pressure, absolute pressure in the alveoli of 750? I'm not going to shift up atmospheric pressure on you. All right, cool. All right, so 750 is now lower than that atmospheric pressure of 760. So we are now breathing in, sucking air towards that low pressure environment. So let's do this how we would like to do it in our relative pressures now. Have a relative pressure in the alveoli of -10. You breathe in or out. So for relative pressures, we've set atmospheric pressure to 0. Okay. So anytime we have a negative number, that's telling us we have a low pressure environment in the alveoli. So this is the exact same example as the previous question. We've just changed it to be a relative pressure. So we're still breathing in. So we're 10 millimeters of mercury below that 760 atmospheric pressure. We're entering the lungs now. Okay, so one more. Got a relative pressure of 15. You're going to have air entering or exiting the lungs. Okay, perfect. All right, so now we're all on the same page, right? Relative pressure means we set atmospheric to 0. So if we have a number above 0, we are breathing out. If we have a number below 0, we're breathing in. So that's why we like to use relative pressures rather than atmospheric, because that's just a little easier to get our minds around. But all we're doing when we're figuring out inspiration and expiration is comparing those two numbers. I'm repeating that to you a couple of times because I hate when at the end of the unit, somebody's getting tripped up by additional information that we learn after today. This should be the easy pit. We're just picking which number is bigger, and that's it, right? So it's going to move away from the bigger number, go towards the smaller number. Cool. How much of it? All right. So one of the pressures that might trip you up, right? Which is why we're starting with just alveolar pressure and atmospheric pressure, right? So the rest of these pressures are important, but don't define breathing in versus breathing out, okay? So our next pressure is our intrapleural pressure. Yeah. So this is our pressure inside that pleural sac, so that lining of the lungs, basically the outside. It's actually always negative under normal conditions, which is great because this is what's going to help us kind of follow the chest wall. You can think of it as like kind of suctioning the lungs onto your ribs. Okay. So that's basically what this intrapleural pressure does. So at rest, it happens to be -4. Okay. So the reason it's negative is that what's happening with the lungs when we have recoil, like your lungs want to collapse, annoying, but they do, right? So the lungs, the edge of the lungs are pulling in. And your ribs kind of want to expand. So your ribs are kind of pulling out. Those are their directions of recoil. So that's what's creating that kind of negative pressure in that space. And that's why we needed some intrapleural fluid in there. So like, it's not just water, but you can think of water, like water creates like surface tension and stuff. We're basically using that intrapleural fluid as kind of glue so that the lungs follow along with our ribs, along with the chest wall. So we are just talking, right? We had our pressure in the alveoli. Now we're looking at rest, right? So if we have an alveolar pressure of 0, right? That means it's the exact same as your external environment. So that's when you're like between breaths, right? No movement of air in and out. Okay, so that's our alveolar pressure. We'd still have an intrapleural pressure at that point because you still want your lungs in contact with that chest wall, right? So we've got that force there. They drew their arrows the other way, that's fine. Yep. That chest wall, right? Oh, this is why. So they drew those arrows the other way because we have the lung moving in, chest moving out. So we need this force here to keep them together, right? And then we have our external pressure, our atmospheric pressure of 760 here. When we're creating relative pressures, we've subtracted 760 out. So we're at 0, matching that alveolar pressure. Okay. One of the reasons this intrapleural pressure is important, one of the places you can imagine this going wrong, right? We're starting out, right? Your lungs want to recoil in, your chest wants to recoil out, but they're blocked by this intrapleural space, this pressure in there, that negative pressure holding them together. Okay. Right. Nice and suctiony. Right. This works so long as you have no gaps. Right. If you get stabbed or shot or something like that, right? If you create an opening between that intrapleural space and the atmosphere, now we have a new pressure difference that we would have to think about, right? We've got that negative pressure in the intrapleural space, and we know that our atmospheric pressure we've set to 0, right? So now we've got an uh-oh situation, right? So we're going to go towards negative pressures, right? So you are going to suck air into that intrapleural space now. If you fill that intrapleural space with air, you don't have room for your lung anymore. And also, we're allowing it to recoil like it wanted to. Okay, so this is how we create a pneumothorax. So pneumo, I think it's a Greek root, has to do with air, right? So we end up with air in the thorax and a collapsed lung here. Okay. You could end up with other things in other situations in this intrapleural space. There shouldn't really be much space here under normal conditions, but you can also imagine it getting like filled with blood or something if you have injuries, right? So if there's stuff in this space, no good. We don't like that, right? So in the second image, we've come to equilibrium between that intrapleural space and the atmosphere, right? Because air will move until we're at equilibrium, right? And that equilibrium point means we've got lots of air and not very much lung tissue in contact with the air that should be coming down your trachea and into those alveoli. So this is a bad situation. That's why we want a negative pressure in that space, but also to make sure we have a closed off container, closed off environment, because otherwise the system doesn't work. Okay. So that brings us to the transpulmonary pressure. Okay, so we're calling this our distending pressure across the lung wall. So we're comparing pressures in the alveoli and in that intrapleural space. So if our transpulmonary pressure increases, basically, this is going to cause your lungs to expand, increasing the volume, and then dropping down the alveolar pressure as a result. So let's think about that. So we got an airway, we got another airway. Okay, so we got our lungs, we've got our intrapleural space. We're drawing that sac and it doubles up, which is why we have a space. Okay, cool. So what we do here, we always have a negative intrapleural pressure under ideal conditions. So we're going to be subtracting a negative, right? Okay, so we're subtracting away -4, right? And let's say we're at rest here, right? So our normal situation at rest is we have 0 minus -4. It'll be a transpulmonary pressure of 4. right? So that's enough pressure to kind of keep that lung tissue kind of glued to your chest wall. Okay. If we are changing that interpleural pressure, right? If it goes down, we're going to see an increase in that transpulmonary pressure, pulling those lungs basically harder. Okay. Cool. Okay. So for our mechanics of breathing, we're moving air in and out of the lungs due to pressure gradients, specifically that pressure between our external environment, so our atmospheric pressure, versus the pressure in the alveoli, that PALV. Our other pressures have to do with musculature, the rib cage, the interpleural space, and their goal is to keep that lung suction to the chest wall, so suction to the inside of the ribs, so that when the ribs expand, your lung expands, the ribs go in, your lung shrinks. That's how we're creating these pressure gradients. Okay. So we'll leave off there and we'll pick up thinking about this flow stuff as relates to breathing on Wednesday.